theoretical reasons. We then looked at cellular ion content when you labeled with different particles. And you can see that with ferrocarbotran residus, you get approximately 10 picograms per cell, which is in line with other published work. And what really struck us in this group was that with this particular particle, M600, we got 3.5 times more label. We didn't believe this at first, so we repeated the experiment a number of times, and we showed that this was in fact a reproducible effect, and that it was highly statistically significant, and that for some reason the other cells, the other particle sizes just didn't get this level of labeling in this particular cell type. So we don't know if this is a cell preference, or whether we were wondering whether this was a cell handling factor, but we think not. We wondered about whether this was an ion measurement problem, so again reproduced this a number of times to show that it wasn't. And we wondered whether this was a specific issue with particle preparation, so we resynthesized at this size and showed exactly the same phenomenon. So it seems to be a size-specific effect, which has not been previously described. Cell viability was preserved and unchanged over multiple passages. We showed that as the cells proliferated, the ion loading went down, regardless of which particle was used, which is exactly as you would expect. Uniquely again, we were the first group to do gene expression profiling using an Affymetrix gene chip to try and see what genes were up and down regulated, and this is a subset. With ferrocarbotran, which is clinically approved, as I mentioned, there are a number of genes that are up and down regulated, mostly ion binding genes. What we observed is that more genes were up and down regulated with M600, but we don't know what this means. Presumably this is related to the ion binding. What we don't know is whether this downregulation of certain cell cycle genes has any significance. Just anecdotally, well, not anecdotally, a couple of papers recently have suggested that nanoparticulate labeling of these cells actually increases the proliferation of differentiated tissues in the labeled cells. We didn't observe this phenomenon in our research, but my current student in Singapore, who is working on a different particle with residus and in mesenchymal stem cells, adult mesenchymal stem cells, tells me he's observing increased proliferation in the chondrocyte differentiation tissues after labeling. So, maybe this is significant. Anyway, what we found was that these particular cells, whether they were labeled with M600 or ferrocarbotran, showed no difference in their trilineage differentiation, no difference in viability, as I mentioned. And then we thought we'd take this into an in vivo model in stroke in rats. So this is actually a well-established model in the brain, and it's using a photothrombotic stroke where we inject the animal with intravenous rose Bengal while shining a high-intensity focal spot of light on a point in the brain through a hole in the skull via a light guide. And this produces a very reproducible cortical stroke, which we can see on the MRI as a high signal area. Then labeled stem cells were implanted with two methods, either by direct implantation into the contralateral side using a very slow injection over 10 minutes of 20,000 cells, or intravenous delivery of 2 million cells, which were followed by a flush. And all of these animals, we could not get aphtharmic rats in our center, so we used cyclosporin immunosuppression, which had previously been reported to be appropriate. These animals were then imaged again with a clinical MRI scanner, not a research MR scanner, and these are fairly typical acquisition parameters. So just to show you what happened. Okay, so here is M600 cell labeling. We can show this is the immediate implantation site, and by day 5 we are seeing labeled cells appearing in the opposite side, at the site of the stroke. Remember, this is the stroke site, and by day 12 this is more intensified. When we use ferrocarbotran, which is a less magnetic particle, less strongly magnetic particle if you like, again we see the injection site, and a few days later we're starting to see it, but it's not nearly as visible as the M600, which reflects just the amount of iron that's migrating there. More excitingly, when we inject it intravenously, even though we know that many of these cells end up in the reticuloendothelial system, the lungs, and so on, we did get cells appearing at the stroke site in 5 days and at 12 days. If there was no stroke, there was no migration. So this tends to validate the fact that, in fact, there is something about the stroke site that attracts these cells to go there. So, superficially, this looks great, and this, in fact, looks exactly like a number of experiments that have been replicated that were published earlier, which we were just copying this model. So this was very encouraging for us. So then we started doing cell staining to see what was actually going on. So we used fluorescence microscopy with various cell labels, anti-rat antibodies, anti-human bimentin antibodies, because we were injecting human cells into rats, so we could try to differentiate the ultimate cell fate. And then we incubated them with additional secondary antibodies. And we also did fluorescence in situ hybridization with looking with starfish probes, and looked at these for nuclear counter staining. So what we saw then was at the site of the stroke on day 1, there was no iron oxide label, both on routine histopathology and with the vimentin and ED1 stains. You could see there was a mixture of human and rat cells at the site of the injection. On day 5, at the stroke site, there was visible iron oxide label, seen by these dark particles here, and that you could see a mixture of human fetal mesenchymal stem cells and macrophages. And then by day 12, disappointingly, there was plenty of iron label, but there were no longer any human fetal stem cells visible by optical microscopy. So this was disappointing. We could clearly demonstrate then that there was migration of the injected cells to the contralateral stroke site, but that these cells were not visible 1 week later. When we mock labeled, we saw no label going across to the stroke site, which is not a surprise. And at the site of the injection, we saw plenty of human cells. When we labeled ferrocarbitran, we saw the same migration again, but in this case, at day 12 again, we saw no human cells. And when we used, and this is the same image I showed you before. So at day 12, whether we label with ferrocarbitran or whether we label with M600, we don't see human cells visible at the site of the stroke, only macrophages. So the iron that we see, we infer, is in macrophages, not in the injected cells. Okay? But that's at day 12. At day 5, definitely human cells. So, well, I've already covered that. So let's move on. So is this cellular rejection? Certainly we saw no evidence of lymphocyte infiltration or any other cellular rejection mechanism that we could detect histologically. And this has been described as an inflammatory kind of rejection. It may not be a human-rat cell interaction because it's been described even in allogeneic rat MSC transplantations. So what we showed, I think, is that this is an effective cell labeling agent. It is 3 times more effective than the clinical agent ferrocarbitran. It reduces the signal 6 to 7-fold over that of ferrocarbitran, and that, at least in our hands, a minimum of 23 labeled cells, but in practice more like 1,000 cells, would be detectable in vivo. The key points is that iron oxide nanoparticle labeling generally appears to be a readily achievable, fairly safe technique from an in vitro standpoint. We certainly saw no safety issues in our injected rats. We can use clinical particles or a variety of other particles. These particular particles appear to be non-toxic, can be tracked. Obviously, the fate of the stem cells needs more research. It's been published recently in Stem Cells and Biomaterials. And then the other members of the group who are more working at the particle development phase in other departments, chemical engineering in particular, have published a number of articles looking at combined magnetic fluorescent particles and particles that evade macrophage capture and so can be more efficiently used for intravenous labeling. And this sort of work cannot occur from a single group. We did the MRI, the chemical engineering people developed new particles for us, the surgery was through pharmacology, the cells came from ONG, we did material science and bioengineering. So this took me about 5 years to assemble this group. I hope it won't take so long again to do it in Sydney. We could do this research in Sydney, and we've already started a collaboration with Macquarie University to start looking at this in a cell population they're interested in. We don't have cell labeling laboratories in radiology, but we do have MR scanners and the experience of working with animals and phantoms and even patients, of course. The problem now is that the particle supply is now constrained. Certainly was much more than it was even a couple of years ago. So other groups are developing their own particles. Veridex is still available. Banks particles are available just off the shelf from Sigma, from the Banks company. Labeling optimization needs to be developed for each cell type you're interested in. And this new technique, again, sonar operation, appears to be very interesting for more efficient labeling of cells in the future. So thank you.
So are there any questions? I'll ask a quick question. Is it that the cells have to be clustered together in order to detect them by MRI, or can you have a diffuse distribution, or does the efficiency of labeling increase?
Yeah, good question. The fact is that, as you said, the single voxel of MR is very large relative to the cell. So the question is, how few cells do you need in that voxel to detect it? This is dependent on two things. It depends how much iron is in the cell, and it depends on the noise level of your image. Now, clinical MR scan is quite sensitive enough to detect these particles, but the noise in the image is relatively high. If you use a high-performance animal scanner, dedicated rodent instrument, you can get the noise floor very low. And in that scenario, the NIH has shown that they can detect one Banks particle in one cell. They've confirmed that histologically. So they've demonstrated that they can see a stem cell implant 1 year later. If there was, provided there was one cell with one particle left, they could still detect it. But this is obviously not something that the average team could do. I think in practical terms, we're looking at 1,000 to 100 cells as the limit of detection.
I'd just like to thank you, Professor Wang, for an excellent example of interdisciplinary successful research. And